from Senegal. I'm the actual membership secretary of the African Society of Human Genetics. AFSHG is supporting the African Pharmacogenomics Network and also supporting this webinar series. APN is now officially affiliated to the AFSHG, and we are currently discussing the possibility of dual memberships for AFSG and APN members. So welcome to this first webinar for the African Pharmacogenomics Network. This is aimed to showcase pharmacogenomics research in African population. We hope many of you will be inspired to develop research proposals in pharmacogenomics. This webinar series will run monthly from now until February 2024. So today is the first webinar, and I'm very pleased to welcome you all and really honored to introduce Professor Collet Dandara. But before the introduction, I just would like to say that Professor Dandara is a professor of genetics at the University of Cape Town. He is fellow of the African Academy of Science and the Academy of Science of South Africa. He is a principal investigator in the Pharmacogenomics and Drug Metabolism Research Group and director of the Platform for Pharmacogenomics Research and Translation of the South African Medical Research Council. He has extensive experience in pharmacogenomics, research and on infectious and cardiovascular disease in African population. He is one of the leading experts in pharmacogenomics on the African continent with international standing in the field. Professor Dandara has a track record of successful supervisions of graduate and postgraduate students with over 10 PhD supervised. He's also widely published with more than 160 publications in international peer-reviewed journals. He is a former board member of the South African Medical Research Council, member of the executive committee of the African Society of Human Genetics, and a founding member of the African Pharmacogenomics Network, which is organizing this webinar in collaboration with the African Society of Human Genetics. So Professor Dandara, my brother, welcome. And thanks for accepting to share with us your experience about pharmacogenomics research in Africa. Before we start, I just would like to give you some ground rules about this webinar. So please type your question through the presentation in the Q&A section. And if you would like to talk during the Q&A session, please raise your hand and I will give you the floor for it. And note that this session is being recorded and will be available via YouTube. So Professor Dandara, you have the floor. Thank you. Professor Collet Dandara. You are muted. Professor Collet.
Great. Hi, Rokhaya. He seems to be having a bit of a technical issue. So we'll just give us 1 minute and we'll try and resolve that. Thank you.
Okay, thank you.
Hello.
Prof. Dandara, could you maybe exit and then rejoin, please?
So we are very sorry for this technical issue, but we hope that in a few seconds, Professor Dandara will be able to give his talk. So all our apologies for that.
Colleagues, it's a great pleasure. Sorry for the technical problems. Rokhaya, I could hear you clearly. Thank you so much for the introduction. I'm glad to be here. Due to time, I think I want to dive straight into my presentation, then we can have discussions. Let me share my slides.
Thank you, Prof. Collet.
Now, can you see my slides, Rokhaya?
Yes, absolutely.
Lovely. Thank you, colleagues. It is really a great pleasure for me to present on this platform, particularly on a subject that I so much have passion for. Today's presentation will mostly be looking at the theme of trying to build capacity and networking in pharmacogenomics across the African continent. So I will get you through my presentation, but I will be trying to answer three main questions of who are we as African researchers, as Africans, and as African pharmacogenomics interested people, as well. Why should we be bothered about pharmacogenomics? And also, what should we do if we are bothered? So I will take you through, and at the end, then we can have a discussion. I'll try to be quick. So if you look at us as we are as part of the global community, we need really to understand who we are in order to appreciate what ought to be done in pharmacogenomics. Some people will tell you that there's a lot of work that is happening in the US, there's a lot of work that is happening in Europe, but I will show you that it is important that work in genomics is also done on the African continent, on Africans. If you look, Africa is the home of humanity. Humans originated from Africa. Africa exhibits the greatest genetic diversity that you can see. If you look, most of the population, populations across the world originated from Africa, but only a fraction of the populations migrated out of Africa, leaving Africa with the greatest diversity. If you look into the peoples of Africa in terms of phenotype, you will see actually a cross-section of different phenotypes that actually speaks to the underlying genetic diversity. This has been confirmed by work that we've done over the years, which has actually shown that when you characterize sections of the genome or across the genome, and you compare populations of European, Asian, and African populations, you will find out that they will isolate into the different groups. However, when you further analyze the African population group, you will find out that it is hugely diverse. So it is in this huge diversity that is often missed when genomics is used in health, because the fraction, most of the trials that are done in terms of drug discovery are done either in European and Asian populations, and they miss a lot of the diversity that we find in African populations. If you look in terms of disease burden, if you look at the total cause of disease burden, if you look and compare African populations, you will agree with me that Africa has got a different disease profile when you compare it to either European and American populations. The African populations actually have got largely an infectious underlying disease burden, with an increasing non-communicable burden, which actually is different from when you look at Europe, which has got mostly a non-communicable disease burden and very little of the communicable disease burden. This is quite very important in the field of pharmacogenomics, because when we look at pharmacogenomics, we are looking at the intersection between the drugs that we use for the treatment of this disease and the underlying genetics. So already you start to see that here we are premising who we are in terms of the profile of the diseases that we encounter in Africa compared to the rest of the world. Breaking it further down, you can actually see that the top diseases that you find mostly in African populations are not the same as those that you find in the Northern Hemisphere, which already shows you that even on the genes that are going to be targets for treatment, if genes that are going to be the sources for enzymes to metabolize drugs, we are at times looking at different targets. So this profile is very important when you talk about pharmacogenomics, because now we want to really make sure that the work that we do is speaking to the likely drugs that are going to be used in the African population. Here I give you some of the data that we've generated over the years, which shows you just a few enzymes that metabolize some of the drugs that are commonly used in treatment. Here I'm showing CYP2C19, CYP2D6, N-acetyltransferase 2, GST, M1 and T1, and CYP2B6. These are the enzymes that metabolize most commonly used drugs. Below them, I'm showing here the variants that can happen in these genes. And these variants actually affect the activity that it can be obtained from an enzyme that is made from each of these genes. What I want to draw your attention to is that when you look at the populations here, the top populations here are non-African populations. While it's those from here, we have included the African Americans. You see populations of African origin. What is important is to see that when we look at variant distribution, the mutations that happen in these enzymes that are so important in the metabolism of some of these drugs, you will find that for some variants, for example, CYP2D6 star 17, they are not found in populations that have got, let's say, distant African ancestry. Whereas you find that that variant is quite common in the populations of African ancestry. So this profile of difference can actually also exhibit itself in the way that when patients who are of European and African origin are given drugs that are metabolized by CYP2D6, their ability or their profile to interact with that drug is affected by whether they have got star 17 or any of the variants that is differing between these populations. One reason why we must also study African populations, because when you, for example, with star 17, when you study non-African populations, you miss to pick the effects of star 17 because it is not found in those populations. Going further, if you were also to look in terms of if you were to look in terms of disease burden according to age, when you look at compare Africa to Europe, you will find that most of the disease burden in Africa, up to 55%, is actually for the age group 5 to 14 years. If you compare with Europe, it means that most of our young population is actually affected by disorders. If you were to push it to 15 to 49, you're actually taking more than 80% of the population to be affected by that huge burden of disease. So another difference also that we have a difference in the profile of which particular age groups of patients are affected in terms of the diseases. This is very important because most of the drugs that are developed, the clinical trials are mostly done in European populations, which means they are trialed on adult or older populations, yet they are going to be used particularly to treat diseases that are prominent in young populations, giving us also another idea that we must make room for trying to include younger populations as we carry out clinical trials. Here, I'm just showing you per capita that is lost due to communicable and non-communicable diseases in different parts of the world. You will see that the disease burden in Africa hits the young. It actually hits the age groups where you actually need those people to be the pipeline for the workers. You will find that we lose quite a lot. In most of the African countries, you find them here, that there's a huge loss in terms of per capita, in terms of the rehabilitation of the diseases compared to the rest of the world, where actually they've got a huge per capita. So we have low money, but also we are affected mostly by diseases. So that is actually also very important as we put this into perspective. Now, I wanted to bring you back to pharmacogenomics in terms of looking at how does then pharmacogenomic interface with therapeutic drug use. There's not much coherent data for the rest of the world, but the US does collect very good data that you can look at in terms of how therapeutic drugs are used. Here I'm presenting to you data for 2015 to 2018 in the US, where they're actually looking at the use of drugs, prescription drugs in the US in that period of time. What you will see is that there is a huge use of drugs. In a period of 30 days, a month, you find that nearly 50% of people in the US use drugs, therapeutic drugs. It's a huge number. And if you take those nearly 50% of the people, about 25% actually are on 3 or more prescriptions, giving us actually the importance of also pharmacogenomics. But of those also, you identify that even about 13% are on 5 or more prescriptions. And we know that drug-drug interactions is a huge issue, and pharmacogenomics is one of the pillars that can actually help us tease out which are the drugs that are likely to interact so that we can use alternative drugs. Now, if you go back to the magnitude of the number of people affected by communicable diseases, which is the huge component in African population, you will see that we almost above 300 million. And at some stage we had to hit above 400 million, meaning that a huge percentage of our population, young population, is affected by disease, being driven by huge drivers like HIV, malaria, and other neglected disorders. This is just to show you that all these particular individuals would actually need to be under therapeutic treatment. But don't forget that each of those individuals has got different profiles of pharmacogenes and might require different drugs for them to have quality of life. And here I'm showing you also now what you can see in terms of the distribution of the burden with age. But here I've just brought all the major drivers of some of the infectious diseases that we've in this part of the world. HIV, look at how much Ebola has ravaged this part of the world compared to the rest of the world, and also malaria, a perennial problem that we face. But interposed to that is the constant throwing in of drugs or therapies that we try to contain these medications. So if you, just to reiterate one of my earlier points, that most of the drugs that you find now being used across the world are developed through clinical trials in Western countries. But I've highlighted that the age groups of people affected is different, the underlying genetics is different, the diseases that are in different places are different. So we really need to up our game as African researchers, that we must pay attention, we must move with the rest of the world so that we can also start to contribute to understanding the improvement of quality under therapeutic treatment for patients. It will be remiss if I don't mention sickle cell disease. Sickle cell is one of the leading diseases, causes of mortality in the children under 5 and the youth between 5 and 14. And to make matters worse, I've shown you how much disease burden we have in Africa. Given that across the world, 500,000 babies were born with sickle disease, nearly 80% of these are actually in sub-Saharan Africa. So this is also compounding on top of the communicable disease burden. Sickle also gives another huge burden on the peoples of Africa. So what we then need to do is, for us, I've shown that we have very little in terms of gene per capita, in terms of funds. We are not privileged that we can just research on anything. I think with the scant resources that we need, we don't have time to waste. We need to really focus on impactful research so that we can make a contribution to our populations. If you look in terms of disease burden, the panacea to disease is treatment, but before you get the disease, I think one of the ways is to work on prevention. In terms of pharmacogenomics, we already dealing with individuals who have already been infected or affected. So under treatment, the huge problem that pharmacogenomics is likely to solve for us is the problem of adverse drug reactions, which then leads to quality response if we can identify those individuals who will not do better on a particular medication and those who will do better. So again, I refer to data from the US, which is well curated, where you find that it is estimated that between the fourth and sixth common cause of actually in the world is of death is adverse drug reactions, and they actually cause a significant mortality and morbidity burden in vulnerable groups such as the old people, children, and particularly also people in low-income countries, because we can't afford some of the interventions. So adverse drug reaction-related health costs are significant. In the US, they are estimated to cost about 30 billion per year, and in Europe about 79 billion euros. We are yet to also do our calculations in Africa. I think we've got data for particular hospitals, but not for entire nations. So although ADRs can never be entirely eliminated, they can be significantly mitigated, reducing both financial costs and human suffering. So this brings me to our next one of the findings that we did, where we looked at efavirenz was at that time the most commonly used drugs in the treatment of HIV, and the standard treatment was 600 milligrams per day, but it was given as a one-size-fits-all. And what happened was that you could identify patients who responded very well, and some who in the middle, and some who did not respond well. Yet among the three groups of patients, the virus was taken care of, but the quality of life was only high in the group that did not have the mutation. So we worked on this and said efavirenz is one of the aspects about efavirenz is that it is an effective drug, also quite affordable. So we went on with a colleague, Professor Masimire in Zimbabwe, to develop what we call the gene dose intervention, where we propose that actually you can actually give efavirenz after you have identified what genotype an individual is carrying, and then propose that they are given different doses according to their genotypes. So by the time we were successful in showing this, that this is possible, efavirenz was taken off the market, because a better drug had come, which is dolutegravir, DTG. But I want to make a point here to say dolutegravir it's more expensive than efavirenz. We might even stop using DTG for newer medications, but I think this is evidence where you can show that using pharmacogenomics, you can actually rescue cheap drugs that are effective. given that the population that are targeted, especially for HIV, are populations with very low per capita. So that's one of the issues that we must work on pharmacogenomics to rescue those drugs that pharmaceutical companies might start wanting to stop to manufacture because they are saying they've got side effects. If we can show that we can identify or predict who is likely to be affected before dosing and keep those drugs in use. I want to quote a colleague of mine, Professor Charles Rotimi. This is a quote that he wrote, which is written on the African Academy of Science in Nairobi. So I was passing by, where really his sentiment speaks to what we are discussing today. That we really the diversity of the African people in terms of their underlying genetics is important. If we understand that genetic diversity of African people, it will help us really to rescue the rest of the world in terms of identifying biomarkers of disease susceptibility as well as treatment response. So as a bit of history, I want to take you back to 2003, when the first draft of the complete human genome was released. There was a very important meeting that happened in Nairobi, Kenya. You can see me here, well, I was still very young there with the rest of the continent. We were very ambitious. We had our first African pharmacogenomics meeting in August of 2003. And the idea was we could really see that we really needed to be part of this journey to decode the human genome so that it can be helpful in improving the human health. If we jump to further years, then we come to 2019, we met with a group of colleagues again in Nairobi, and we came up with an open letter that was to be now the impetus for us to form the African Pharmacogenomics network that now we're starting to move with. And then COVID came and everything almost disappeared. So we are now reinvigorating this spirit that started in 20 years ago. So if we have to take a reality check in Africa in terms of pharmacogenomics, we know that we have got fewer expertise with the genomics knowledge and even fewer with pharmacogenomics knowledge. And it is difficult also in Africa to access genomic platforms, and there is lack of understanding of pharmacogenomics stakeholders. So really we need to build this capacity. We need to come together to really work together so that we can be part of answering the questions using genetics. I will take another quote from one of my very inspirational leaders, Kwame Nkrumah, who on the eve of Ghana's independence in 1957, he proclaimed that our independence is meaningless unless it is linked up with the total liberation of Africa. I want to borrow this, mutate it as a geneticist, and say, so is our individual research success, unless it is integrated within the rest of Africa and the rest of the world. So, as the African Pharmacogenomic Network, our strategic plan is pinned on 5 pillars. We would want to establish centers of excellence. Already we have got places where pharmacogenomics is being practiced, where students are being trained. We want to establish a pharmacogenomics clinical trials platform. Remember, I said most of the drugs are being trialed in populations that have different underlying genetics, also different ages. We want also to argue that we must even start to reduce the age of people who can be admitted in clinical trials for the benefit of the patient population. We also want to foster networking and resource sharing, and we want to develop pharmacogenomic guidelines that are helpful for the African continent and push with advocacy activities. So those are the strategic plans for APN. But I would want to move to support that. The reason why we're taking this approach is the realization that we know that we don't have much money that every individual can buy an Ion Torrent or a Novoseek. But we can make use of fewer platforms to have impact. There is no use for each individual to buy a Novoseek and only use it once, because we cannot afford the next batch of reagents. So that is the Cralion core. We are not going to compete with the rest of the world in terms of how many Novoseeks we have. We're going to use the fewer that we procure to make an impact as in Africa. Now, if you look on what we our current activities. So our current activities is that actually the first one is today. We have started with the webinar series that will be running until February. We will pick them up again as we go. And then the next, we're actually in the middle of seeking funding to support some future activities. We are very, very intentional that we really want to realize our vision. We also want, as part of our activities, to support research grant proposals. We know, I think there are a lot of people who don't understand what does what makes a proposal, a pharmacogenetics proposal. Sometimes people just characterize a few genes there and then already they are coining pharmacogenomics and all that. So we want to be a resource as APN to support people who are doing their individual researches there. If they've got questions, then we help them so that they build strong proposals. The benefit for African Pharmacogenomics Network is that once they have published, that data becomes very useful to the African continent. We also want to have a platform to share resources, like I was saying, that we have got, we don't have to all buy Novoseek or whatever, but we need to know that I can access the resources that are in Strathmore in Kenya. I can access the resources that are in Cameroon and South Africa and so forth and so forth. We are in the middle of a membership drive, so that we continue working together, sort of interacting and influencing each other and supporting each other as we grow as a community. And this also is part of our engagement with stakeholders, but we will roll it more as we want to talk to our physician colleagues as well as the general public in terms of the utility of pharmacogenomics in the broader realm of genomics in health. So those are the current activities that we have, but we are not ending there. We also very ambitious. We are looking forward to develop a short course which has got which will have modules. I must hasten to add that although we have not yet concluded firmly with AGMT, we admire the way they've run their modules, and really we would want to see if we can sort of use work with them as a support base as we roll out these modules. The first one will be an introduction to pharmacogenomics. We want basic training in terminology and concepts. Then we'll go to research designs in pharmacogenomics, databases and resources. We'll do that. And then we'll look at how to report pharmacogenomics research studies and presentation of proposals. We are looking at universities that would want to partner with us so that we can make this as an accredited course. So we are very, very ambitious, and please, we hope we can get partners who will move with us in this journey. Yeah, I'm just highlighting some of the students who have trained over the years in the pharmacogenomics field and training for across the African continent. This is not all of them. I just picked a few of them that are there. So to show that actually we can do it, because we are doing it. But I think with more support and working together, we can actually bridge the gap where we can have more expertise in pharmacogenomics or genomics in general. And I really would want at this stage also to acknowledge colleagues who I've worked with a lot in terms of my journey, but in terms of really genomics and pharmacogenomics, particularly Colin Massimilian Ambrose there, Michelle Skelton. We've started working together with Mohammed. Also the work, also the collaboration we're starting with the Faznu, and Vicky comes with a lot of also expertise, and you say who we work with in the different projects as well. So these are not the only people who are important in pharmacogenomics, but we are building our base, and we have got a membership drive that we are doing, and we hope to continue growing strong. So I would also want to thank the technical team, although they were keeping me out in the beginning of the talk, but they managed to bring me in. Thank you so much, Mario and Wilson, for creating this platform and assisting us as we kick off this journey of trying to build capacity in pharmacogenomics. So I really would want to thank you. But before I do that, I want you to encourage you to join APN, and you can use that website, and you can, if you've got any questions, you can actually send them via email to that email address. So I would want to leave time for questions. So I really appreciate. Thank you very much.
Thank you very much, Professor Collet, for this wonderful and inspiring talk. So it was really interesting to see that there is a lot of work that is undergoing in Africa about pharmacogenomics. And as you raised during your presentation, the genetic diversity of the African population is so high and not yet studied. So we need to study our population to define the profile of drug metabolism in order to set up new drugs that fit to the African population. So that is very interesting. And you show that there is a lot of challenges that we have to face. And we have a lot of young researchers that will face through this challenge. I know that you are very engaged in supervising students, graduate and postgraduate students. So I'm sure that in the following years, we will have a good team of African researchers in pharmacogenomics to tackle these problems about all infectious disease that have a huge burden in African population. So now we can open the Q&A session. I got one question in the chat from Emilienne Tata. So she said that, thanks, Prof, for the presentation. So I think this is a contribution. She said that the pharmacogenomics is not directly linked to sickle cell gene therapy. So what do you think about that?
And interesting, remember, the genes that we call pharmacogenes, they can play a role that is either activating or inactivating. So if you're thinking of gene therapy, sometimes we might even want to go in there and correct that gene so that you have got a working enzyme. So it's not entirely divorced. You can actually also employ gene therapy on pharmacogenes, but usually most of the genes that you get in pharmacogenomics, if they are alternative drugs, you better just getting alternative drugs than getting the correction of the genome.
Yes, yes, absolutely. Another question is from Piasa. He's one of my students. He said, as we know that African population are underrepresented in genetic studies and databases, what are some potential implications and benefits of including African-specific data in pharmacogenomic research?
Lovely. So one of the things that you can see, I will refer to the work that we did, and it was led by Professor Masimirembo, where he was looking at just exposure to drugs that are metabolized by CYP2D6. And when you looked for all the variants that were reported in Africa, in European populations, you then assign a phenotype and say, this is a fast metabolizer. But when he assigned fast metabolizer status to African populations, their phenotype in terms of how they were breaking the drug was actually poor metabolizer, which means there were still some variants in the African population that were not captured in the European population. So the importance of studying African population, that we will discover more variants that are still hidden, that have not yet been reported, because we've mostly studied populations that are not as diverse as African populations. That's the importance of it.
Okay, thank you, Professor Collet. I have a question.
Yes.
And my question is about CYP2C8.
Yes.
That is associated with drug metabolisms used for malaria.
Yes.
So what are have you worked on that? And what are your experience about the mutational profile in African population? I ask this question because in a small study that we started in our lab,
yes,
we see that the allele 3
yes,
of CYP2C8
yes,
is very quickened. The frequency is about 50% in Senegalese patient with adverse reaction
yes.
to the use of ACT. What do you think about it?
You are very right. So we just graduated a master's student from Ethiopia who was resident doing his work on pharmacogenomics here in our lab. So he just finished his master's, he's completed. He looked at CYP2C9 and other genes in terms of their contribution to the pharmacogenomics of antimalarials. And yes, we pick some variants actually that make individuals who carry those variants, or most of the time you actually see that they report adverse effects. So we have not yet published that data, but we will package it. Yes, it's an important gene in terms of anti-malaria metabolism.
Okay, thank you, Prof. So I will follow up with you.
Lovely.
Thank you. Another question from Rokhaya Ndiaye. So what role can early career researcher play in strengthening the success implementation of the APN? Is there a way of starting a committee lead by early career research to support the main organizers of the APN?
It's a very important question, Arinao, and congratulations, Arinao. She graduated last week with her PhD on the pharmacogenomics of warfarin. She has now taken a postdoc with one of the major funders we have here. Yes, I think it's not even to support this. We starting these initiatives. Young emerging researchers must actually come and take them over. That's the call. We are using our connections and all that. You come and take over and run them, because you have got a different agency. And it will be very interesting for those who are interested to come in and say, this is our work, we want to do it. So you can't be relegated to supporting. This work is ours together, and particularly the young researchers. So yes, APN is calling. Let's work together. Come in and push what you want to see happening, so that we grow this society. But what you must also look at is, one of the things we're doing is we are applying for funding. We want the funders to be able to support us so that we can retain these young people in Africa here doing the research in Africa. Because the table that I showed you of those PhDs, I think half of them are already in the US because we cannot retain them. So that's one of the things that we want to do. Or sometimes they are frustrated that they have learned so much, but they can't do anything because there's no money to do their work. So if we work with centers of excellence, we can be able to support each other so that quality work is still done, so that you can start to grow as a young researcher and you don't have to go to out of the continent. Thank you.
Thanks, Professor Collet. You are absolutely right. I think that, as we say, Africa cannot develop without Africans. So young, in particular in research, young researchers have to be in Africa and work on the topics that are of interest of the African population. So thank you for that. I have another question. It's from Emily and Tata, the first question. So she said she was asking if sickle cell gene therapy has been implemented in Africa.
Come again. What is the question?
She said if sickle cell gene therapy
has
been implemented in Africa.
No, not that I know of at the moment. I know there's a lot of work that is happening, and also if you look, one of our supporters for one of our groups, or colleagues who work with Professor Honkam and others, they've got a huge network on sickle cell. I must say I'm not sure where sickle cell gene therapy is, but I know it's at advanced stages, but I've not heard that we have started it in Africa. Rokhaya, you can help me on that one. Do we have any, do you think it has been rolled out in Africa? No, I don't think so.
No. From the last discussion I had with one of my colleagues working on sickle cell disease, they didn't start yet. But I think that there is a lot of researchers in Africa that are interested in this topic, and maybe in few years we may start, because you know that the profile, the genetic profile of the mutation in sickle cell disease is very diverse within the African population. So even if there is a gene therapy, it could not cover all the variants that are involved. So maybe this is going to be done step by step, taking into account specificity, geographic specificity of the population. Another question is about one of my students, because I asked my master degree student to join the webinar.
Yes.
To complete their knowledge and skills about pharmacogenomics.
Yes.
So she asked about how to validate the pharmacogenomic studies in terms of ethics.
Okay, great.
I think specific ethic committee, or
so in my institution here, we still use the same, what we call the Human Research Ethics Committee. That's all the human research, they can handle that. It caters for genomics in general. So that's the committee that you can use. And I think the committees are now learning from each other. It was difficult in the beginning, but it's becoming easier and easier as they get information from others.
Okay, and another question from Manasi Kilonzi is, are there rapid tests for pharmacogenomics? If yes, are they available and affordable?
That's a very, your question, Kilonzi, is very important. Greetings, Kilonzi, and I'm hoping that you are going to join us here in Cape Town from Tanzania. So I'm very much looking forward to that. In terms of rapid tests, yes, if you look at the UPGX, a huge trial that was done in Europe last year, where they did pharmacogenomics across about 15 countries, they actually used rapid tests. But if you look at cost, on the costs in terms of using the Vocare, they're very expensive. So what we are advocating is that once we know which variants we want, we will use it in what we call a preemptive manner, where you come to the clinic, for your first interaction with the doctors and they prescribe a drug, you get that drug, we take your blood sample and we genotype you for the panel of important variants. But from the second time until the rest of your life when you come, we already have your pharmacogene profile. So that now we can be mining from that information and treating you using your information. So we only miss the first visit because our characterization might take longer and you need to be treated then. But should you come again, then you are already covered. The results are in your electronic records and they can be used, again and again and again. That's the way we're thinking about it.
Thanks, Professor. So the last question, I think will be from me about the APN. So how are you managing to involve more African researchers in this network?
Lovely. We are very purposeful. So what we encourage, if you look at the opinion paper that we published, it's people from across Africa who are on that paper. Now we've got major centers who are training people in pharmacogenomics. We are encouraging people to recruit from outside of their countries. I can mention one program that Professor Masimire in Zimbabwe was doing, where he came up with a program where he's training masters in pharmacogenomics. And he takes about 10 students and only 2 come from his country. The rest are coming from across the continent. That way, I think it's a model that we can use to really support. And when they're finished their masters, some of us here, we are in South Africa, we absorb them for PhD. So we're training, using the various capabilities that we have in the continent so that we increase this capacity that we so need in pharmacogenomics.
Okay, thanks. But in terms of collaboration, do you think that through grant application?
Yes, that's a very good one. We are actually in the middle of scouting for grant applications. We're looking at for possible responsive calls that we can respond to as a consortium. So we will bring in people. We have invited people we will invite people, but currently we're just building the sort of the objectives that we want and looking for the appropriate cause. But yes, we want it to be an APN grant. So we are busy also trying to register APN the proper way so that it can be able to receive those grants, and then we can really do the work that is possible.
Okay, thank you very much, Professor Collet. It was really nice to see you today and have this wonderful talk. And it was my pleasure to moderate this session. So
thank you very much.
And thanks to all the participants. And I would like also to thank Vicky Nembore for her assistance, Wilson and Mario for the technical assistance. So thanks to you, and hopefully the next series, the next webinar in a month, we hope. Thank you very much, and have a wonderful evening.
Thank you.